Bismillah ar-Rahman ar-Rahim. This is the third lecture in virology, and it will be about the diagnosis of viral infections. In this lecture, we concentrate on the laboratory diagnosis of viral infections. The laboratory diagnosis of viral infections based on three general approaches. First approach, it will be the direct detection of viral components, either in cells derived from infected tissue or free in fluid specimens. Second approach is the isolation of viruses. Usually, this is done in cell culture, followed by identification of the isolated virus. The third approach, done by demonstration of a significant increase in serum levels of antibodies to the etiological virus, or demonstration the presence of antibodies of class IgM specific to etiological virus. This is the three approaches used in laboratory diagnosis of viral infections. Certain points should be mentioned in a specimen collection in viral infection. First thing is timing. Specimens for virus isolation and direct detection, as well as blood samples, must be collected within the first few days of the illness. The other point is choosing the specimen. This is done by knowing the pathogenesis of the virus. A good example for this is poliovirus infection. Here, the neurological samples is not important in the diagnosis, but the throat wash is important because the virus starts replication in the oropharynx. So by knowing the pathogenesis of poliovirus and the virus enter through the oropharynx and start replication in the throat, by knowing this, the choice of specimens for the isolation of the poliovirus is by taking throat wash. The other point in specimens in viral infection is clinical data. The specimen sent to the lab should contain clinical data indicating the type of virus that the physician looking for, in order to the lab worker can choose the special type of tissue culture, because each virus need special type of tissue culture to support its replication. So clinical data is important. Now, specimen transport and storage. Specimens should deliver without delay, keeping the specimen at neutral pH. This to avoid affection of the viral infectivity. Keeping the specimen at 4 degrees centigrade if delay is expected for 4 days by refrigeration. If the delay in shipping of the specimen to the lab is more than 4 days, the specimens should kept at minus 70 degrees centigrade, freezing. Some virus is liable to freezing. Here, samples should be diluted with an equal volume of 2 concentration saccharose in phosphate buffer, the pH 7.2. Culture and isolation. The culture and isolation is remain the gold standard in viral diagnosis. Viruses do not grow on artificial media. They must be cultured on cell culture or other type of living system. From this living system we have 1. Embryonated eggs. Here the egg we use is usually hen egg. Fertilized hen egg should incubate it for 5 to 14 days in order to be embryonated, and after that inoculation of the embryonated egg can be done. The site of inoculation and the period of incubation is depend on the type of the virus. After the inoculation and incubation, the embryonated egg examined for the evidence of viral replication. Evidence of viral replication in the inoculated embryonated egg can be on form of 1. Death of the yolk sac. This appears as shrinkage in the yolk sac of the embryonated egg, or it appear as pox formation. This is cytopathic effect on the chorion of the embryonated egg. Or simply we can examine the fluid of the embryonated egg looking for viral antigens. This picture showing the embryonated egg and that there is a different site of inoculation depending on the type of the virus. So we can inoculate the specimen at chorioamniotic membrane or at amniotic membrane or yolk sac. This depends on the type of the virus. This picture showing the pox. Pox formation is a visible foci or lesions on the chorioamniotic membrane of the developing chick embryo. This is the pox. This cytopathic affects the pox. It could be coarse as in the upper right B picture, coarse pox formation on the chorioamniotic membrane of the chick embryo. This is the cytopathic effect of vaccinia virus, where in the upper left picture A, the pox formation, the cytopathic effect of variola virus on the chorioamniotic membrane, it appears as a fine scattered pox formation. The second form of living system using laboratory animals. This was used in the past. Mice, chimpanzees were inoculated with the viruses, but now this has been largely replaced by using of cell cultures. Now we will come to cell cultures. Cell culture originated from viable host cells and grow into a monolayer on the basis on the sides of a glass or a plastic test tube. So we take a viable host cell, manage to grow as a monolayer, and this monolayer put on the sides of a glass or a plastic test tubes. This viable host cell in the monolayer must be kept moist and supplied with the nutrients. This is done by keeping them immersed in cell culture medium. So the monolayer of the cell cultures keep immersed in a cell culture medium. This cell culture medium provides the cell with the nutrient. Every now and then, these cells must be maintained, washed by maintenance medium. The importance of maintenance medium is to neutralize the effect of toxic product due to the viral replication in these cells. So the cell culture, it will be a combination of a monolayer of viable host cell grown on a size of a glass or plastic, test tubes, flask, etc. Plus moisture and nourishment of these cells is done by cell culture medium, plus maintenance of the medium done by maintenance solution that done every now and then to neutralize the toxic effect of the waste product. Cell culture incubated from 1 to 4 weeks. This is depending on the type of the virus. Periodically, the cell of the cell culture are inspected for the presence of the virus. The presence of the virus in the cell indicated by areas of dead or dying cells. These dying cells called cytopathic effect, CPE. Cell culture used or help in 1. Isolation and growth of a variety of viruses. 2. Studying viral replication. 3. Development of attenuated mutant strains and vaccine production. This is the use of cell culture. Isolation of the virus, studying viral replication. 3. Development of attenuated strain of the virus and vaccine production. This picture showing the process of having the monolayer on a flask or a glass test tube. We managed to take animal, adult or embryo or other living system like egg. We take the tissue from this living system, dissecting the tissue, then treating these with diluted trypsin enzyme in order to have enzyme digestion. Finally, chop of this tissue in order to have a cell grown as a monolayer, as a thin monolayer. After that, this thin monolayer grown on a sides of glass or plastic, flask, test tube, microtiter plates. This picture in the upper right, the monolayer, the viable host cell, managed to grow as a monolayer after the dissection, enzyme treatment, and chopping. We have the monolayer, and this monolayer we can put it on different kind of plastic or glass flask, petri dishes, or microtiter plates. This picture to remind you that the tissue culture or the cell culture is not only the monolayer viable host cell grown on a flask or petri dish or microtiter plate. This cells need to be maintained and nourished. This is done by having this cell moist with cell culture media and maintained every now and then with a maintenance medium. So this picture of a refrigerator containing the cell culture medium and the maintenance medium. Now we have a term, a cell line. A cell culture becomes a cell line once it has been passed. This means subcultured in vitro. So once the cell culture passed subcultured in vitro, it will become a cell line. Cell lines are classified as primarily cell lines, low passage cell lines, or we can call it secondary cell lines, and the continuous cell lines. The primarily cell lines, those cell lines have the capacity to pass only once or twice in vitro. Further passages of these primarily cell lines result in decrease the receptivity of the cell lines to the viral infection. So in the primarily cell lines, these cell lines have the capacity to be passed, to be subcultured once, twice in vitro. After that, there will be deterioration, decrease in the receptivity of these cells, of the cell lines, to the viral infection. Example of these cell lines, we have the human embryonic kidney, guinea pig embryo, chick embryo, and the primarily monkey kidney. These are examples of primarily cell lines. These cell lines can be passed only once or twice in vitro. After that, it will be deteriorated, decrease in the receptivity to the viral replication. The second type of cell lines is the low passage cell lines. Those cell lines that remain virus sensitive through to 20 to 50 passages. Low passage cell lines remain sensitive to the virus, receptive to the virus through 20 to 50 passages in vitro. Here we take diploid fibroblast cells such as lung fibroblast, commonly used in low passage cell line. So the examples for the low passage cell lines we have: 1. A human diploid fibroblast cell line, MRC5, an institution of medical research, and the third is the WI38. Now we will come to the continuous cell lines. Those cell lines usually prepared from malignant cell and can be passed and remain sensitive to the virus infection for indefinitely. Continuous cell lines prepared from tumor cell can be passed subculture in vitro and remain sensitive, receptive to the virus for indefinitely. Example of the continuous cell lines we have HIP2, it's a human epidermoid cancer of the larynx, and the second example is HELA, cervical carcinoma cells. For the identification of the viruses in cell culture, in order to identify the virus that grow in the cell culture, here the infected cell in the cell culture change their morphology and eventually lysed or detached, that is meant to elevated from the surface of the glass during a while dying. These changes in the infected cell called cytopathic effects of the virus, the CPE. So the CPE is a change in the morphology, elevation from the surface of the glass of the cell culture while the cell is dying. All these changes is called the cytopathic effect of the virus. Viruses are detected in cell culture by recognition of these cytopathic effects. Viruses have distinctive cytopathic effects, just as colonies of bacteria on the agar plate. CPE, the cytopathic effect, can be quantitated on the cell culture. Now, the quantitation and the interpretation. If we have no change, no effect on the cells of monolayer, this is negative. If there is atypical change in the morphology involving a few cells in the monolayer, this is equivocal, plus minus. Need to request another sample. A +1, this means that 1 to 25% of the cell in the monolayer exhibit cytopathic effect. A +2, 25 to 50% of monolayer cells exhibit cytopathic effect. +3, 50 to 75% of the cells in the monolayer exhibit cytopathic effect, while +4, more than 75% of the cells in the monolayer exhibit the cytopathic effect. Now, viruses detected in the cell culture can be identified by 1. Knowing the type of cell culture that support the viral replication, what type of the cell culture the virus replicate in it. 2. The time need to detect the cytopathic effect, how much time need for the cytopathic effect to appear in the cell culture. And 3rd, the morphology, the kind of the cytopathic effect that appear in the cells of the monolayer. By combining these 3 points, type of the cell culture support the replication of the virus, time needed to the appearance of the cytopathic effect, plus the morphology, the kind of this cytopathic effect, we can identify the virus grow in this cell culture. Morphology, now we will come to the morphology of the cytopathic effect. This cytopathic effect is a suggestive of infection by a family, but do not allow us to determine the species level of the virus. Example of the cytopathic effect. A. Herpes viruses, herpes viridia family, all the herpes viridia family viruses cause cytopathic effect in form of loss of architecture plus rounding of the cell, and the appearance of this cytopathic effect, it will be appear after 24 to 48 hours of the inoculation of the cell culture. B. In case of paramyxoviruses, this include measles virus, respiratory syncytial virus, cells coalesce into a large multi-enucleated syncytia, and this appear in 3 to 5 days. Syncytia formed by membrane fusion of the infected and the non-infected cells. And I have a picture after this slide of the large multinucleated syncytia formation. C. The cytopathic effect of cytomegalovirus, the CMV, it will be in form of appearance of foci of cell lysis in 2 to 3 weeks after the inoculation of the cell culture. And these foci of cell lysis, it's called the plaques. Again, I have a picture after this slide for the cytopathic effect of the CMV virus. D. Viruses may cause the appearance of inclusion bodies. Viral inclusions, they are intracellular structures formed by A. Aggregates of viruses within an infected cell. B. It could be abnormal accumulation of the viral components from inefficient viral replication. Inclusion bodies are visible by conventional microscopy. Now, the location and the appearance of inclusion bodies provide information about the infecting virus. Example, we can take a stained smear from base of a skin vesicle used to detect inclusion bodies of varicella zoster virus or herpes simplex virus. This test is called the Zank test. This is slide showing the syncytia formation. Syncytia is the fusion of cells into large multinucleated cells. This is the cytopathic effect in respiratory syncytial virus and measles virus, because the infected cell with the virus lead to the expression of viral fusion proteins on the surface of the infected cell. Now, this infected cell, due to the viral fusion proteins, will be fused with the uninfected cell, and the another infected cell lead to the formation of multi-nucleated syncytial cell. This slide showing the inclusion bodies. In the upper, nicely spindled uninfected host cell with a clear nucleus in the center. In the lower right picture, we have cell infected with reovirus showing a perinuclear inclusions. The inclusion in a cell infected with reovirus appear as a crescent surrounding the nucleus of the host cell. It's called perinuclear inclusions. Whereas in the lower left picture, cell infected with cytomegalovirus with nuclear inclusions plus cytoplasmic inclusions. This slide showing the cytopathic effect of CMV on a human embryo skin muscle cell culture. The A picture showing uninfected cell with the virus. B, it will be a late cytopathic effect of CMV virus in form of a nuclear inclusion, the clear black dot inside the nucleus of the cell of the tissue culture, plus enlargement of the cell. In the C, we have a cell degeneration, a clear lysis of the cells of the monolayer appearing as loss of architecture with cell degradation and degeneration. If we go at low power visualization of the cells of monolayer in this slide, it will be appear as a focus of cell lysis in cell monolayer, This is called the plaque formation. So in this slide, we have the cytopathic effect of CMV, beginning with the enlargement of the infected cell with intranuclear inclusions, followed by cellular degeneration, and lastly the appearance of a foci of cell lysis on a cell monolayer, what we call the plaque. For definitive identification of the virus grown on a cell culture, we have a battery of tests. Tests that can be used for definitive identification of the virus grown on the cell culture. First test, we can take the fluorescent labeled antisera. Fluorescent labeled antisera. This is available for most viruses and used for culture confirmation. Second, tests used for definitive identification of the virus grown on the cell culture. We have the viral neutralization test. These tests used to identify viruses with many serotypes for which fluorescent labeled antisera are not available. Third test, we have the acid liability test. This test used to differentiate between enteroviruses, which acid is stable, differentiate these viruses from rhinoviruses, which acid labile virus. Fourth test is the heme adsorption test. Some viruses, like the influenza virus, parainfluenza virus, mumps virus, can be detected by the heme adsorption test, since the infected cells with these viruses contain viral heme adsorbing glycoprotein in their outer membranes. The fifth test used for definitive identification of the virus is the electron microscope or the immune electron microscopy to detect the virus inside the infected cells in the cell culture. These 5 tests used for definitive identification of the virus grown on the cell culture: fluorescent labeled antisera, viral neutralization test, acid liability test, heme adsorption test for some viruses, and the 5th is the EM electron microscopy or immune electron microscopy. Now we come to the shell vial method. Shell vial method, a rapid modification of a conventional cell culture. Here, virus is detected more quickly by staining host cell in the cell culture for viral antigens before the development of the cytopathic effect. In shell vial method, the monolayer of the cell culture is grown on a cover slip. This is the shell vial, and the specimens are inoculated onto the shell vial cell layer by low speed centrifugation. This low speed centrifugation, while inoculation taking place, enhance the viral infectivity. So the shell vial method: A, shorten the time of the virus to get in the cell. B, enhance replication of the virus. C. Decrease the time of the cytopathic effect of the virus to occur. Shelvial method used for viruses required long incubation period before producing cytopathic effect. Example, CMV need 4 weeks to show cytopathic effect in conventional cell culture, while by using Shelvial method, a few days required for cytopathic effect to appear. This slide showing the Shelvial method as a modification of conventional cell culture. Here we have a centrifuge tubes with cover. In the base of these tubes we have a cover slip, the Shelvial, where the monolayer cells manage to grow, and where the inoculation of this monolayer on the cover slip on the Shelvial taking place while the specimen under low speed centrifugation. This low speed centrifugation enhance the viral replication and shorten the time of the virus to enter inside the cell and decrease the time of the appearance of the cytopathic effect. Now, we have a rapid diagnostic test. Rapid diagnostic test used in medical virology. These tests, in comparison to cell culture, which required a few days, here a few hours required after receiving a specimen, we can detect the virus. So we call this test a rapid diagnostic test. A few hours required after receiving the specimen and the result can be there. These tests include 1. Direct electron microscope examination of the specimens. Example, examination of the stool for rotavirus and hepatitis A virus. 2. Immune electron microscope. This allows visualization of virus particles present in number too small for easy direct detection. 3. Antigen detection method. The antigen of the virus is detected by immunofluorescent technique. Example for this test is the detection of the respiratory syncytial virus antigen in the respiratory secretions. 4th, agglutination method. Here antibody is added to the specimen to detect the presence of the viral antigen. 5. Molecular technique depending on a nucleic acid hybridization, used to detect viral genome and genome sequence in clinical specimens. In molecular technique, we have A. In situ hybridization technique. Use single-stranded complementary nucleic acid probes. It is highly sensitive and specific. This technique used to identify A. Adenovirus in nasopharyngeal wash. B. CMV in urine. C. HIV in the blood of seronegative individuals. B, for example of molecular method, is the PCR. PCR increases the sensitivity of a nucleic acid hybridization by increases the amount of viral genome obtained from the patient. This approach is useful for rapid detection of the virus in patient specimens when the amount of viral nucleic acid in patient sample is small. 6. We can use serology. Serological test used to detect antibodies produced against viral antigens. From these serological tests we have 1. Radioimmune assay, RIA. 2. Enzyme immune assay, ELISA. 3. Indirect immunofluorescence test. 4. We have the heme agglutination inhibition test. Measure antibodies directed against hemagglutinating viruses such as influenza virus. 5. We have the complement fixation test. So in serology, we can use radioimmune assay, RIA, enzyme immune assay, indirect immunofluorescence test, heme agglutination inhibition test, and lastly we can use the complement fixation test. All these serological tests aiming to 1. Detect a significant increase in the titer of antibodies to the etiological virus. This can be done by taking 2 samples, 1 sample at day 0 and measuring the amount of antibody to the etiological virus, and repeat the test after 2 weeks in the second sample, looking for a significant increase in the titer of the antibodies. A significant increase in form of 4-fold increase in the titer. This significant increase in the titer indicating that the serum of the patient is having the antibody to the etiological virus. The second aim of the serological test, the presence of IgM specific antibodies to the etiological virus. So in the serology, we are aiming for 2 things. 1. Looking for a 4-fold increase in the antibody titer to the etiological virus. This is done by taking 2 samples, sample at day 0 and the second sample after 2 weeks, looking for 4-fold increase in the antibody titer, or by using 1 sample looking for a presence of IgM specific antibodies to the etiological virus. By this we are covering the most important laboratory test used in medical virology. The last slide for the hemagglutination inhibition test. In hemagglutination inhibition test, we use the RBC as a reagent and the viral particle coated with the hemagglutinating antigen also as a reagent. So if we have the RBC plus the hemagglutinating virus plus the serum of the patient containing no hemagglutinating antibody, the result will be hemoagglutination. While if we add the RBCs plus the serum of the patient which containing antibody directing against the hemagglutinating antigen on the surface of the virus plus the virus, there will be inhibition of the hemagglutination. This is the principle of hemagglutination inhibition test. This is only for the knowing, not included in the test. And thank you.